All right, now this is the second series of the glycolytic pathway. I don't like to rush it at all. I need to break it down. So if you not watch the first video, please do well to go and watch the first video so that you understand where we are coming from. Now we have taken a good breakdown of what glycolysis is and where it occurs and which segments it occurs in the cell. Now this is where we stopped in the last video, and remember I told you that this DAP that is formed, now I'm calling it DAP now, dihydroxyacetone phosphate. Remember, this is 3 carbon. Show you the same. Why this one is also what? 3 carbon. But the thing here is that this compound here is not stable, so you have to be converted to what? Glyceride diphosphate, which is the simplest monosaccharide, glyceride diphosphate, which is a 3 carbon sugar that is being phosphorated in carbon number 3. Now that is not it. Now remember, I also said something in the series in course of this video that most times, especially if you are writing an exam that have to do with theory, that you need to be drawing these molecules properly so that you get the full mark, because normally you need to draw the structure of glucose, structure of glucose phosphate, fructose phosphate, and which is very, very easy. It all depends on the examiner what they ask you for. Maybe they can say give the glycolytic pathway using the physical projection, or they can say use the Howard projection. Now look at what I'm trying to say. Use the physical projection, or the Howard projection. Now I'm going to be bringing other videos concerning this aspect, but please let's take a rundown of the food pathway. Remember, we have taken only 3 steps: exokinase, glucose to glucose phosphate, glucose to fructose phosphate, and fructose phosphate to fructose 1,6-bisphosphate. Then of course, now this is the 4th step, now talking about the breakdown, the cleavage of these to form this. Now what happens next is that, of course, like I said just now, that these will not proceed to the pathway now. So let's continue from that aspect very, very quickly and very, very fast. Now remember that we have what? Glyceraldehyde. No, would they take them, they go. Glyceraldehyde. So make I take them like this. Glyceraldehyde, 3 phosphate to save time. Now, remember now that we are having 2 molecules of glyceraldehyde 3 phosphate already in the pathway, because one DAP has been converted to glyceraldehyde 3 phosphate. So we're making it 2 molecules of glyceraldehyde 3 phosphate. Now what now happens is that this glyceraldehyde 3 phosphate is now converted to what? To 1,3-bisphosphoglycerates. 1,3-bisphosphoglycerates. Show you the same now. Now, which enzyme catalyzes this reaction? And this reaction is a redox reaction. That you must put at the back of your mind. The conversion of glyceraldehyde 3 phosphate to 1,3-bisphosphoglycerate is what? Is a redox reaction, and it's catalyzed by what? By glyceraldehyde 3 phosphate dehydrogenase. Show you the same now. Now this is the enzyme. So please, for those of us that are new to enzymology, talking about enzyme and its activity and their names, dehydrogenases usually catalyzes redox reaction in metabolic pathway. So most time when you see this name dehydrogenase in the enzyme name, just know that that enzyme is categorically catabolizing or breaking down or is involved in redox reaction. Now come back to me now. Now this is the enzyme that catalyzes this reaction, and in that process NADH is formed. Show you the same now. NADH is formed. Now this is the only NADH that is formed in this pathway. Now remember that anything that is going to be happening from here down is going to be happening in 2. That means it's 2 molecules of 1,3-bisphosphoglycerate that is formed. It's 2 molecules of NADH that is formed. Why? In the sense that it's 2 glyceride diphosphate 3 phosphate that is being broken down. Show you the get them now. Now from this place, another thing I need to say here is that this is the first energy containing compound that is formed during glycolysis. Please jot that down. This is the first energy containing compound that is formed during glycolysis. So this guy is an energy containing containing compound. It's an energy containing containing compound. That is this guy here. Now from 1,3-bisphosphoglycerate, this guy is now broken down to form 3-phosphoglycerates. Very good. 3-phosphoglycerates. Now, this is the first step that brings about the formation of ATP. Now what happens here is that you can see here that we're having two phosphate group. So one of them is what is donated to what, to ADP, to now form what, ATP. Now if you listen to the video, what I said just now, you notice that I said that this reaction is happening in what, in twos. Show you the get am now. Therefore, it's two molecule of ATP that is formed here. It's two molecule of 3-phosphoglycerate that is formed here. Show you the get am now. Now, there's one other thing I need to spotlight, but maybe not in this video, in subsequent video. So please do well to subscribe so that you get to see videos like this and other videos. Now, there is an intermediate molecule that is formed before 3-phosphoglycerate, known as 2,3-phosphoglycerate, bisphosphoglycerate, and this is usually formed in RBC, red blood cells. Talking about the Bose effect and all that. Now we're going to break down these videos for you so that you get to understand this series of videos with Mosjaro. Now let's continue. Now in this place, of course, it's a kinase that catalyzes this reaction. Now that kinase, 1,3-phosphoglycerate, what kinase? Now if you hear an enzyme with the name kinase, like in the first enzyme, that first regulatory enzyme known as exokinase, it involves the transfer of what, of phosphate group. Now this phosphate group is being transferred from this high energy containing compound to ADP to now form what, ATP. Now from 3-phosphoglycerate we now have what, we now have 2-phosphoglycerate. Now nothing specific is happening in this particular step, and this enzyme, the enzyme that does this reaction is a mutase. What does it do? A mutase just carry the phosphate group in carbon number 3 to carbon number 2. Show you the get am now. From here to here is what is just a mutase carrying this phosphate group from carbon number 3 to carbon number what, 2. Now what now happens next is that this 2-phosphoglycerate is now converted to what, to phosphoenol pyruvate. Phosphoenol pyruvate. Now this phosphoenol pyruvate now is the second energy containing compound, PEP, which is also known as what, PEP. You are going to come across this during photosynthesis, the Calvin cycle, C4 plants, C3 plants, the C3 plants. Now look at it now. Then from this place we now have what, we now have the what, the pyruvate or the pyruvic what, acid. And in this reaction is being catalyzed by what, a kinase again known as what, pyruvate kinase. Why this one is catalyzed by what, 3-phosphoglycerate what, kinase. This one is catalyzed by 3 pyruvate what, kinase. Now this is the third regulatory step in glycolysis and is catalyzed by pyruvate what, kinase. In this step, ADP is converted to what, to ATP. That means when that ATP is formed here, that means 2 ATP is now formed here. Then 2 ATP here. So in this pathway, we use 2 ATP to form what, 4 ATP. So what's the total number of ATP produced during glycolysis is 4. But if they now ask you what is the net ATP produced during glycolysis is what, 2 net ATP. That means in the calculation, I produce 4 ATP, not Russian. Now I produce 4 ATP. But remember, in producing this 4 ATP, 2 ATP was used to produce the what, the 4 ATP. So if I pay back my 2 ATP, so what the net gain is what, is 2 ATP. So be very careful when you're asking the question, what's the net ATP produced is 2, but what's the total ATP produced in glycolysis is what, 4. So I use 2 to manufacture 4 ATP. So what the net ATP that I've gained is what, is 2 ATP. Very easy. Now you have known already the committed step and the regulatory step. The committed step is in the first one that I did just now, the first video, which is what catalyzed by the PFK1, phosphofructokinase 1, which is the one that catalyzed from fructose 6-phosphate to fructose 1,6-bisphosphate. Now that one is a committed step. The first regulatory step is reaction number what? Reaction number 1. The other regulatory step, which is the committed step, is reaction number what? Reaction number 3. Show you the same so. And this other one now, show you the get am now. So these are the reactions. So we have 3. So reaction number 4, then we have 5, we have 6, we have what? 7, we have 8. Show you the get am now. Then we have what? 9. Now, ladies and gentlemen, of course, this one is 6 because 6, yes, 6 because we have to convert gap to what gap. This is 6, this is 7, this is what? 8, this is 9, this is what? 10. So, glycolysis is a 10-step process. Very easy. And in this 10 step, we have 3 regulatory step and one committed step. So please, you must pay attention and go through the video and know those enzymes that catalyze the regulatory step in what glycolysis. You must know the committed step. You must know the net ATP produced. You must know the total ATP what produced. And of course, remember, these NADH also produce here 2 NADH, which is a reducing what equivalent. Now remember that we stated from the beginning of the video that glycolysis occurs in the cytoplasm. Now after this pyruvate has been formed, it has other fates. Now this pyruvate, listen, no, this pyruvate is channeled into the what? Into the TCA cycle, but not directly. Is first converted to what? Is first converted to what? To acetyl-CoA. The reason being that this pyruvate cannot enter into the mitochondrion, cannot cross the cytoplasmic mitochondrion barrier. So it's first converted to what? To acetyl what? CoA. And what enzyme catalyzes that reaction? That reaction is catalyzed by the enzyme known as what? Pyruvate what? Pyruvate dehydrogenase complex. It's a complex of enzymes. Pyruvate dehydrogenase what? Complex catalyzes this reaction. Now you are seeing a dehydrogenase here again. That means this reaction is a redox what? Reaction. That means it forms what? It forms NADH. And that one is formed here. We need this information because we need to calculate the number of ATP produced in respiration generally. That means both glycolysis, the TCA cycle, or oxidative phosphorylation. Another thing you must know again is that one is that theoretically, one NADH in the electron transport chain of this phosphorylation is synonymous to what? To 3 what? ATP. While one FADH2 that is formed during the TCA cycle is synonymous to what? 2 what? It's equivalent to 2 ATP. Show you the same now. So we need to have that information. That's why I need to put this at the back of our mind that NADH is also produced in the glycolytic pathway. Now, this is one phase that can lead into the TCA cycle, and of course, we're going to be looking at the TCA cycle and the important significance of the TCA cycle, just like we did for that of what 8 glycolysis. Now, this pyruvate too in muzzle, when there's absence of oxygen in the muzzle, can form what? Lactate or what? Or lactic acid. An accumulation of this lactate or lactic acid results to what? To muscle pull, or what you call what? Muscle cramp or muscle fatigue. Sure, you understand this matter now. Now remember, what can inhibit the pathway? What can inhibit the pathway? IATP can inhibit glycolytic pathway. Not forget them. IATP can inhibit what? Glycolytic pathway. What can also inhibit glycolytic pathway again? Please, high amount of citrates can also inhibit glycolytic pathway. Another thing that can inhibit glycolytic pathway again is what? Is high amount of what? Of what's it called? High amount of glucose. Sorry, high amount of glucose stimulate glycolytic pathway. So you can see all these breakdown. So what inhibits, what stimulate. So see what we will do now. We could just break them down. Sharp time no day. Now please pay attention to this. What inhibits glycolytic pathway? I said ATP inhibits, citrates also inhibit glycolytic pathway. Show you the same now. What stimulate glycolytic pathway? High amount of ADP stimulate glycolytic pathway. Sure, you get them so now. Glucose 6-phosphate also stimulate glycolytic pathway. Sure, you get them now. But remember, insulin and glucagon has role to play in what? In glycolytic what? Pathway. Low insulin and high glucagon. They say so. Low insulin and high glucagon inhibit what? Glycolytic pathway. But high insulin increase glycolytic pathway. So insulin and glucagon has a role to play when it comes to what? Glycolysis. So you have fully seen the glycolytic pathway. You have understood the glycolytic pathway, the steps involved, the enzymes involved, how is it inhibited, how is it what? Regulatory step, committed step, number of ATP produced, what stages produce the ATP, and the enzymes that are what? Involved. Now, in the next video that we're going to be looking at when it comes to carbohydrate metabolism, we want to look at the TCA cycle. Now, when this acetyl-CoA enters into the, remember, the TCA cycle does not occur in the cytoplasm, it occurs in the what? Mitochondrion. And that TCA cycle occurs in aerobic condition, unlike glycolysis that occurs in anaerobic condition. So our next series of video, we're going to be looking at the TCA cycle, the steps involved in the TCA cycle, Show you the get am now. And we're going to looking at the reducing equivalent that are formed in the TCA cycle. What is the significance of the TCA cycle? Show you the get. But please, there's one other thing I want to bring to your knowledge here is that this pyruvate can also generate glucose back in the gluconeogenesis pathway, and that pathway is called what? Gluconeogenesis. Show you the same. So some people say it's the reverse of glycolysis, but it's not totally, because you know why? The steps that are involved in gluconeogenesis are not the opposite of glycolysis directly. Show you the get am now. So pyruvate cannot just go and form also not pyruvate directly. So it forms a molecule first, forms also not pyruvate, and all that, all the steps can go. So we're going to be looking at gluconeogenetic pathway in this series of video with Mosjaro. So ladies and gentlemen, boys and girls, so if you have any question, place it in the comment section. If you know that you don't understand any particular part, please put it in the comment section. You can message me directly, so we'll get to trash it as we move ahead step by step. Remember, we are dealing with what? Carbohydrate metabolism. Now we're going to look at protein metabolism. We're going to look at lipid metabolism. Hope you are getting it now. And again, remember, I promised you a video that we're going to be looking at carbohydrate classification, talking about carbohydrate, non-carbohydrate, and sugar carbohydrate and non-sugar carbohydrate, test for carbohydrate and all that. So please do well to subscribe, do well to share the video, so that other persons can get to see these and other detailed video. Thank you very much. I'll see you in my next video.